everyone, welcome to the interviews of the South American Mycorrhizal Research Network. My name is Camille Triong, and today I have the pleasure to be with Laura Bogar.
Lovely to meet you.
Laura, you recently started a position as assistant professor at the University of California, Davis. Congratulations.
Thank you.
And today we are going to discuss intensely with you that you published in New Phytologist called Modified Source Sink Dynamics, Govern Resource Exchange in Ectomycorrhizal Symbiosis. So, in a general context, what can you tell us about this source sink dynamics and how can we apply this to ectomycorrhizal fungi?
A good question. So in general, I think about source sink dynamics as just the simplest possible way to think about resources moving through a system. I have friends who work in fisheries and they use this concept a lot. And the concept generally just describes any scenario where you have a lot of something in one place and very little of that thing elsewhere. And then the stuff moves from the source to the sink. So perhaps it's a fishing reserve area where there's tons of fish, and then they could move into the sink area where the fishing is happening. Or in the case of plant resources, we think about tissues like leaves being profound sources of fixed carbon, because that's where the photosynthesis is happening. And then a lot of the other plant tissues end up being sinks for those carbon resources. And so I think it remains a really useful sort of a null hypothesis way to think about how carbon should be expected to move between plants and the microbes and fungi that are attached to their tissues. Fungi need carbon, the plant has carbon, and in a very simplistic sense, in some ways the fungi extend the plant root system, and it can be useful to base our predictions of how much carbon the fungi should have on what we think should happen if it were just a sink tissue that's receiving carbon in a sort of passive way.
So basically, the plant is the source doing the photosynthesis, and then the fungi are going to be the sink, right?
Exactly.
But then in terms of nutrient, is it the other way around? Then the fungus is the source and the plant is the sink? Is it going both ways?
I think that's a really interesting question and is one of the reasons that I'm excited about source sink dynamics as a hypothesis to play with, because there should be scenarios where the fungus is kind of a source for these nutrients, especially if the fungus, as it often does, has enzymatic abilities that the plant doesn't have. It can unlock resources that would be hard for the root to get by itself. And in that scenario, the fungus is acting sort of as a source of potential resources, which could then flow into potential plant sink tissues where those resources are needed. It's complicated though, because for carbon, of course, the plant is the only one in that partnership that can fix carbon from the atmosphere. But when we're talking about soil resources like nitrogen or phosphorus or a lot of these things, plant roots by themselves can also access many of these resources on their own. So if we're talking about more accessible, simple forms of nitrogen, for instance, there's no guarantee that at any given moment the fungus will be a more effective source than the plant, except to the extent that the fungus can get in the way of the plant's own uptake capabilities. So it's an interesting opportunity to think about it as potentially a pipeline with a source and a sink, but then there's some valves in the pipeline and twists and turns that might prevent some smooth flowing of resources that otherwise would happen.
So to go back to this like trade between the fungi and the plant, how does this work? Is it like a 1-for-1 molecule? And then does the plant have some kind of control on how much carbon it gives away? Or what do we know about this?
It's such a great question. I think many of us are interested in the answer. I certainly have been fascinated by this for more than a decade. And when I first started thinking about experiments in this realm, I really wanted it to be something like one molecule for one molecule. It's very appealing. It would just be so simple.
Yeah, exactly.
Like I really would like that to be the case. And I think there are some circumstances in which a plant can invest a certain amount of carbon and expect a certain amount of nitrogen or phosphorus back. I think you can create circumstances where that happens. But the more I look at experiments I've done, and especially experiments other people have done, it's so context dependent that I think pretending that it's a X number of molecules for Y number of molecules kind of trade misses a lot of the nuance. There's situations where the investment may be incredibly asymmetrical. Maybe the resource the plant is getting from the fungus becomes incredibly expensive for whatever reason. And that reason may not have to do with the availability of the resource in the soil. It might instead have to do with what else the fungus is getting up to and how strong a carbon sink it's exerting.
When there is more available nutrient in the soil, especially nitrogen, the colonization rate of ectomycorrhizae tends to drop. So do you think that the plant has some kind of control over how much it gets colonized and how much carbon it's giving away?
It's a great question. I think it probably depends a little bit on the plant and the fungus because we have such diversity on both sides. But it does look, I mean, your observation about reduced colonization rates and high nitrogen is totally true. We see modulation of the rates of interaction and to a certain extent modulation of carbon investment, which could track just with number of root tips, and it can also differ across root tips. There's still a lot of experiments we need to do, right? I don't think we have a good answer for this, especially not across host plants or across fungi. It seems likeliest to me that we're going to figure out that plant immune response to the fungi is where most of that control happens, where the plant either allows for mycorrhization, the fungus can get into the root and establish symbiosis, or the plant launches an effective enough immune response that the fungus cannot do that. And there should be some dynamic range between those two extremes where, like, I don't know what the plant is keeping track of exactly, because it could be any number of things that it responds to specifically in the fungi, including it would be very satisfying, again, if the plant responded directly to resource flux from the fungi. I'm sure they also detect more classic sort of chemical crosstalk signs from the fungus that it may be trying to go deeper than the plant wants it to in its tissues. I suspect the plant has some immune control, but otherwise maybe doesn't have a lot of control over how much resource, how much carbon flows into that root tip beyond sort of trying to crank off the symbiosis at all. But I do think it's really interesting because I suspect that some mycorrhizas will be more effective pathways for carbon than others, and it'll depend on the fungus involved and what the plant is doing in response.
So this immune response for the plant, somehow, would you say that this is similar to a plant response to like a root pathogen attack?
I think it might be. I think it would. It should involve some similar genes anyway. It's. I think there's going to be a lot of exciting work over the next few years using both the few ectomycorrhizal host plants that we can actually do genetics with. So, like, I think probably work in Populus will tell us a lot more about what are the specific defense pathways that are involved with ectomycorrhizas, and are they distinct from the specific defense pathways that are involved for standard fungal pathogens or not. But when we do gene expression analysis on somewhat less tractable plants, you end up with annotations in the sort of plant defense sphere. It seems likely that plants are using the same kind of tools that they would use for pathogens when they realize that a fungus is not what they want to have on their roots anymore.
Is it something that you are planning to do in future research now?
I hope to. I think we're going to need to look at this question in many different ectomycorrhizal systems in order to understand how it works more generally and what kinds of responses a plant needs to have established in order to engage in this kind of symbiosis productively. So I'm currently trying to spin up a bunch of projects with little baby Douglas fir seedlings and hoping that the Douglas fir can give me some hint into into what's happening with that host plant at least. But but I'm really curious how that compares to eucalyptus and how that compares to populus, and what it looks like on the whole across these independently involved ectomycorrhizal hosts.
What do you think are the tools that we need to answer these questions? Is it we need more reference genomes for a wider range of ectomycorrhizal hosts, better annotated gene families, maybe?
It's like DougFur is a system that people have used, but most of especially our transcriptome references, do not involve roots at all. And so the critical genes that are happening on the roots, like I am going to have to produce those reference libraries to like understand what genes are involved. So yes, better annotations will be great. That'll be really powerful.
And root transcriptome. Yeah, that's a very good point.
Because I'm excited about the promise of poly-A sequencing approaches and sort of the 3' seq. The possibility of getting a lot more information with a lot less sequencing depth. But in these non-model systems, if you do not have a really strong reference transcriptome, it's not useful.
I see.
So yeah, better genetic resources in terms of the sequencing. It's also true that until we can manipulate either the host gene expression or ideally the host gene content, the molecular plant biologist will not be able to take seriously our assertions that such and such gene is involved in discriminating between ectomycorrhizal partners.
To go back to the fungi in the soil, something that I have always found fascinating is that one tree usually associated with dozens of ectomycorrhizal species from a lot of different groups with different origins, different enzymatic pathways, right? Fungi that obviously also compete with each other. So do you think that these sourcing dynamics could explain part of this complexity?
I think it could. I mean, you just articulated the question that I find most motivating about like this entire field. It's like, why are there so many fungi on a single root system? And this is a thing I especially do like about the Douglas fir, is that like they can have many, many different fungi on their roots. And I've been trying to come at this question from a number of different angles. Like if the plant has a lot of control, maybe through this immunological turning on and off of symbiosis, maybe each of the fungi has a different benefit. And so it's kind of strategic for the plant to have a fungus that is really great at getting nitrogen and a different fungus that's really great at getting phosphorus, or like this other fungus is amazing at fighting off a pathogen. And so if there's a way for the plant to sense all of that and keep track of different dimensions of benefit that it's receiving, then in principle, you could end up with diversity on a single root system. But that does require the plant to have like a lot of control of the situation. I think it's likely that there's some plant control, as we've discussed, and the plant as a carbon source and the fungi as a carbon sink. But also the fungi themselves are serious agents that are controlling a lot of this as well. So the fungi not only vary in their potential benefit to the plant, they also vary in their potential ability to exploit available root space, grab carbon, regardless of instantaneous benefit. So I think variability in fungal traits and the strategies that they use to interact with the plant, and variation in fungal benefit to the plant could all be part of it. And I've also been exploring, speaking of the source sink thing, the idea of more sort of neutral ways that we could end up with this kind of diversity. I'm working with my postdoctoral mentor. She and I were exploring this question together when I was working with her, and then we recently got a grant to keep looking at the possibility that some of this diversity might actually just be sort of plant leakiness that ends up being evolutionarily useful as a way for plants to maintain diversity without having to sense everything that every little fungus is doing. So we know that even if you're just a Arabidopsis plant and you don't have a bunch of fungi on your roots, even then your roots tend to leak. There's root exudates. The roots themselves are a sink for plant carbon. And so we're launching a project right now to try to quantify the extent to which Douglas fir seedlings leak carbon towards portions of their root system that are not very useful in the moment. We're going to experimentally manipulate the plants so that those roots really don't have anything to contribute. Maybe they don't have a fungal partner at all. Maybe they have a worthless fungal partner, and compare where the plant carbon ends up going between that and a much better, more useful fungus. And I'll be interested to see how consistent that leakage is, and how intense it is. If the plant really is just kind of a unscrupulous carbon fire hose into the soil, as long as the fungi that end up on the root system are not so bad, it could still be good through time to have like some fungi that are good and some that are like okay, because maybe they'll be good later.
Yeah, that's a very good point, actually. There is like a spatial but also a temporal aspects to take into account. And I guess finally we end up with more questions that answers maybe here, but this is what research is about, right?
Yeah.
There's a lot of fungi and other microbes in the soil that potentially compete with each other for these resources. So do you think that this competition aspect could be included in the model of source and sink?
Absolutely. I really like competition as almost the most fundamental mechanism of community ecology. It structures everything and produces and incentivizes some cooperative interactions that we see at sort of a higher level that emerges from competition. So yeah, the ectomycorrhizal fungi are fighting each other for space on the roots. They're fighting other soil microbes for those root exudates. Probably this is tied to their ability in part to suppress some pathogens. There's some direct fighting of the potential pathogens and other effects on plant immunity, right? But I do think that the fungal tendency to fight each other should impact the sourcing dynamics as well, because I can't remember the citation. There's excellent papers with the carbon-14 labeling. You can see carbon-14 accumulating at the growing edge where fungi fight each other. And it's easy to imagine that sort of interaction creating a pretty intense carbon sink that may not directly benefit a plant. I would love to see experiments where we could actually compare carbon investment in fungi that are actively fighting something off versus fungi that are being useful partners. And eventually that fungus will eat the fungus that it has killed, and then that's a nice little nutrient source.
Yeah, exactly. Yeah, that's a good end for the story.
Yeah.
Well, thank you again for talking with us today. This is really fascinating research. So you just started your lab recently. Can you tell us a little bit about other area of research that you're interested to dig into in the future?
Oh, I like the verb to dig. We can start there. I'm excited about a lot of different questions. We're setting up the stable isotope enrichment equipment to look carefully at plant resource allocation like before, and hopefully at some fungal resource allocation to the plant. In the field, I'm really excited to start getting into understanding fungal biogeography with depth underneath plant root systems. I've been really interested in the oak savannas out here in California. There's root systems that go down to 8 meters in the unconsolidated bedrock, which I cannot sample right now. That is too deep.
Amazing.
But I do really want to. I've been thinking a lot about a lot of these trees will die. We have increasingly intense summer droughts, right? And I'm fascinated to think about what the oak carbon subsidy does to the fungal and bacterial communities at depth, and what happens when you shut off that fire hose by killing the plant. What difference is this going to make in terms of the carbon cycling in the soils out there? So that's where the digging led me. At the sort of more seedling experiment end of things, I have some students who are really excited about looking at molecular mechanisms of symbiosis, and so they're setting up careful little microcosms with individual Douglas fir seedlings and individual fungi from our culture collection, and getting developmental gene expression time series to understand both what that procession typically looks like in Douglas fir and also how variable it is across different fungi. We've got projects looking at post-wildfire restoration. We've got a lot of drought physiology and gene expression seedling projects happening right now. And of course, we're launching this sort of plant leakiness set of experiments, ultimately looking at whether fungal diversity can serve as a kind of bet hedging for adult trees to survive variable environments through time. So through all of that, it's exciting. There's a lot of directions, but it's great. And if those are themes that viewers are excited about, I'm hoping to recruit a graduate student this cycle to start next fall. And I'm affiliated with 3 different graduate programs at Davis. So send me an email as soon as you can if you're interested in that, because the deadlines are coming up next month. I would like to be able to recruit a postdoc sometime in the next year, although I haven't yet identified the perfect funding source for that. So if that's of interest, folks should reach out to me and we can talk about the kinds of things I'm thinking about and identify opportunities.
If you're interested in Laura's research and want to get involved, please get in touch. Thank you so much again for accepting our invitation and talking about these fascinating topics. And we will be back soon with more interview for the South American Mycorrhizal Research Network.
Thank you so much,
Bye.
Camille.
Thank you.